Okay, so in yesterday's class, we started with the topic of photosynthesis. And we said that photosynthesis is the mechanism through which autotrophic organisms synthesize their own food. So it is an anabolic reaction, which is an energy consuming process wherein the light energy is utilized for the synthesis of glucose or other organic compounds. Now, हमारे पास हमने कल क्लोरोप्लास्ट का स्ट्रक्चर डिस्कस किया था we said that it is a double membranous organelle. It has thylakoid, and a stack of thylakoid is known as granum, and there is a fluid space that is known as stroma. And we also talked about that photosynthesis is divided into two stages. One stage is where the light is required, and therefore it is known as the light dependent stage or the light dependent reaction. And the second part is the light independent reaction, also known as dark reaction, and it is also referred to as Calvin cycle after the scientist who discovered this. We talked about a general summary of photosynthesis, and we would come back to the general summary in just a bit. Then we said that we have thylakoid. Thylakoid are like small, minute, coin-like structures. And upon the thylakoid membrane, we have photosystems, we have electron transport chains, we have the stalked particles. Then we talked about the photosynthetic pigments. We said there are different types of photosynthetic pigments, primary pigments and accessory pigments. Chlorophyll A is your primary pigment, whereas we have chlorophyll B, carotene, and xanthophyll as our accessory pigments. The reason why we have different pigments is because every pigment absorbs light of different wavelengths, absorbs light of different frequency. So, in order to ensure that there is a maximum absorption of light taking place in in chloroplast, we need to ensure that all types of pigments are present which can absorb maximum amount of light. Then we said that these pigments are actually found in bodies called photosystem. And photosystem is like a cluster or a combination of proteins and pigment molecules. It has accessory pigments on the sides or the antennae complexes, and concentrated in the center is your reaction center, where the primary pigments or chlorophyll A molecules are present. And there are only two chlorophyll A molecules that are present in one particular photosystem. I said that the photosystem is like a cluster or a complex combination of proteins and pigment molecules. So we can define photosystem as a cluster of proteins and pigment molecules or a complex combination of proteins and pigment molecules. Now, I'm going to quickly recap with you guys what we did yesterday, as in the brief summary that we discussed of light dependent and the light independent stage. So I want you guys to actively recall with me. Now, can you guys tell me what are the reactants or the starting ingredients of photosynthesis? What do we need for photosynthesis to start? Right. So we have carbon dioxide and water. And carbon dioxide and water need light and chlorophyll, or you can say other pigments, for the synthesis of glucose and oxygen to take place. Now, हमारे पास सबसे पहले light dependent stage होगी या light independent stage होगी? What would happen first? The light dependent stage come first or the light independent stage?
Light
Right,
one.
exactly. So the light dependent stage comes first. And which of the two reactants is involved in the light dependent stage? The water molecule or the carbon dioxide molecule? The water molecule. Very good. So this water molecule enters, or we can say that our photosynthesis starts with the light dependent stage. Now, what happens in light dependent stage is that we are utilizing light, and photolysis of water takes place, due to which oxygen is evolved as a byproduct, and we have hydrogen atoms which split into protons and electrons. And these protons and electrons are essential to produce reduced NADP and to produce ATP. So if when we enter the light independent stage, we would require NADPH, or you can say reduced NADP and ATP. The oxygen acts as a byproduct or a waste product of photosynthesis, and it can either be used for respiration or it can exit via the stomata. Now, once we have reduced NADP and we have ATP, the carbon dioxide can now be reduced to glucose. And this is a brief summary of photosynthesis. Today, we would be studying the light dependent reaction. Now, for the light dependent reaction, we first need to understand that the light dependent reaction is further divided into two parts: non cyclic photophosphorylation and cyclic photophosphorylation. Now, phosphorylation is a term that we all are familiar with from chapter number 12. Can you guys tell me what do we mean by phosphorylation? What is phosphorylation? Addition of phosphate. Very good. So phosphorylation means the addition of phosphate, and over here it particularly means that हमारे पास we are adding phosphate to ADP. ठीक है? And we're also familiar that photo means what? Photo means light, and phosphorylation means addition of the phosphate group. So if we combine these two terminologies, photophosphorylation simply means the production of ATP or the phosphorylation of ADP to ATP due to light energy. So in the presence of light, when we phosphorylate an ADP molecule to ATP, that process is known as photophosphorylation. So it refers to the production of ATP using the energy derived from light. Yes, absolutely, Ahmed. In this case, adding inorganic phosphate using the energy from light. Totally. Okay. Now, this is basically the thylakoid membrane. I would darken it out for you guys. Let's say ये हमारे पास thylakoid membrane है. This is a thylakoid space और बाहर हमारे पास stroma है. Now, this is an ETC, this is an ETC. There are a lot of ETC molecules present on the thylakoid membrane. There are a lot of photosystems present on the thylakoid membrane. There is one thing that is missing in this diagram. Can you guys pinpoint what is something that is missing over here on the thylakoid membrane? Very good. So we do not have a stocked particle over here. So the stocked particle can be present anywhere on the thylakoid membrane. जरूरी नहीं है कि जिस सीक्वेंस में हमारे पास एडीसी और स्टॉक पार्टिकल बने हुए उसी सीक्वेंस में एग्जिस्ट करते हैं. हाँ, this is something that is mandatory, that you need to know which photosystem is placed first and which photosystem comes next. Now, can you guys tell me A and B, which is photosystem 1 and which is photosystem 2? Yes, Amar. I'm going to come to the enzymes once we've discussed the electron transport chain. Correct, Anka. Correct, Amar. Correct, Mutahar. Correct, Ahmed. Exactly. So A is photosystem 2. Why is A photosystem 2? एक तो photosystem 2 पहले आएगा हमेशा. दूसरा, you can clearly see that there is this additional thing at the corner of the photosystem 2, and this is basically your oxygen evolving complex, or you can say the water splitting center. So both of these terms are acceptable, oxygen evolving complex. Now this is a photosystem. This photosystem would have a reaction center, and within the reaction center we would have two chlorophyll A pigments. Now, हमारे पास the light energy is coming from outside, and the light energy is falling upon the photosystems. Let me add the reaction center over here as well. This is your reaction center. These are your primary pigments. Now, the pigments that are present in the photosystems, they absorb light energy. We know that light energy में photons होते हैं. Photons are basically packets of energy. So when light falls upon anything, it contains of these packets of energy, it contains these photons. Now, this light energy is first absorbed by the accessory pigments and then it is transferred to the primary pigments, the chlorophyll A molecule. Now, when the light falls or when the light is absorbed by the photosystems, the primary pigments get photoactivated. Now, what do we mean by photoactivated? Now, when we talk about pigments, pigments are like organic molecules. ये हमारे पास atoms से ही बने हुए होते हैं. Because everything in Earth is made up of atoms. And if it is made up of atoms, it would have the subatomic parts. It would have protons, neutrons, electrons, all of that. Now, when light energy, these photons fall upon the chlorophyll A molecules or the chlorophyll A pigments, these chlorophyll A pigments get excited. They absorb this energy and they get excited. They go at a higher activation level. And when they get excited, they lose electrons. शायद आप लोगों ने फिजिक्स में या फिर केमिस्ट्री में पढ़ा होगा ट्रांजिशन एलिमेंट्स के चैप्टर में या फिर जो आपका एटॉमिक स्पेक्ट्रा का चैप्टर है फिजिक्स का that the electrons can absorb energy, can absorb photon, and they can go to a higher energy level. So exactly the same mechanism happens over here as well. The chlorophyll A pigment absorbs the light energy, it absorbs photons, and when it absorbs photons, it goes into a state of excitation. And when it gets excited, it loses its electrons. And these electrons are picked up by the ATC. So, हमारे पास the electrons would enter the ATC that the chlorophyll A pigment has lost due to photoactivation or due to excitation. Now, the question over here is, हमारे पास बिल्कुल उसी तरह से जिस तरह रेस्पिरेशन में हमने ETC में इलेक्ट्रॉन्स की मूवमेंट देखी थी, the electrons would be moving. They would be moving from one ATC complex to another ATC complex. जो मैकेनिज्म हमारे पास हमने PS2 में ऑब्जर्व किया था बिल्कुल वही PS1 में भी हो रहा होगा. PS1 भी लाइट एनर्जी एब्जॉर्ब करेगा. That light energy would be transferred to the chlorophyll A pigments. The chlorophyll A pigments would get excited, and due to that, they would lose their electrons, and their electrons would be transferred to the ATC. Now, the question over here is, we have studied this previously as well, that the electron transport chain should be continuous, should be in a state of continuous flow. हमारे पास electrons का flow continued रहना चाहिए. क्यों electrons का flow continued रहना चाहिए? Because as these electrons are moving, they are losing their energy. And that energy is used to pump hydrogen ions or protons actively into the thylakoid space. So the energy that these electrons are losing as they move across the ETC, that energy is utilized to actively pump the hydrogen ions into the thylakoid space. Now, if the hydrogen ions are being actively pumped into the thylakoid space, the concentration of protons within the thylakoid space increases, जिसकी वजह से हमारे पास a proton gradient is established. Now we know that stocked particle is also present on the thylakoid membrane. Now can you guys tell me मेरे पास stocked particle का जो ATP synthase है जो उसका head है वो स्ट्रोमा की तरफ पॉइंट कर रहा होगा या थाइलाकोइड स्पेस की तरफ पॉइंट आउट कर रहा होगा? कैन यू गाइस टेल मी व्हाट वुड बी द placement or direction of the stalked particle? Okay, what about the rest of you? Where would the ATP synthase or the head of the stalked particle point? Towards the stroma or towards the thylakoid space? Yes, it would be pointing towards the stroma. Let's say this is your stalked particle. This is your ATP synthase. Now, we have a higher electrochemical gradient in the thylakoid space, and there is a lower electrochemical gradient in the stroma. Second. Okay. So there is a lower proton concentration in the stroma, and there is a high proton concentration in the thylakoid space. This means that the protons can move through the stalked particle via facilitated diffusion. And when the protons move across the ATP synthase down the electrochemical gradient, that process is known as chemiosmosis. And when these protons move through the stalked particle, the energy that is generated during that movement, the electric potential energy that is generated during that movement, is utilized to form ATP by combining ADP with inorganic phosphate. Okay, Shah, I'll repeat this part. हमारे पास बेसिकली यहाँ तक आपको क्लियर है कि इलेक्ट्रॉन्स आर मूविंग अक्रॉस द ईटीसी. इलेक्ट्रॉन्स कहाँ से आ रहे हैं? इलेक्ट्रॉन्स हमारे क्लोरोफिल पिगमेंट्स जो हैं दे आर लूजिंग बिकॉज दे आर गेटिंग फोटो एक्टिवेटेड. व्हेन दीज इलेक्ट्रॉन्स आर मूविंग अक्रॉस द ईटीसी they are losing their energy. That energy is utilized to actively pump the hydrogen ions into the thylakoid space. Now, if the hydrogen ions are being actively pumped into the thylakoid space, the hydrogen ion concentration has increased within the thylakoid space, which means that we have more positive charge and more hydrogen ion concentration gradient in the thylakoid space in comparison to the stroma. This means that an electrochemical gradient has been established. Why is it an electrochemical gradient? Because there is a lesser positive charge in the stroma and there is a lesser concentration of protons in the stroma. So there is a difference in the electrochemical potential between the stroma and the thylakoid. Now, whenever there is an electrochemical gradient established, it is natural that the protons would move down the concentration gradient. Now, when they're moving down the concentration gradient, they cannot directly move through the membrane. They have to move through a channel protein, through a facilitator. And the stalked particle, uski jo tail hoti hai, that is a channel protein for protons. And thus these protons, when they're moving down the electrochemical gradient, they move through the stalked particle. Now, हमारे पास जब भी कोई charged body move करती है from point A to point B, आप लोगों ने physics में पढ़ा होगा that whenever a charged particle or a charge is moving across a circuit, electric potential energy is generated. हर charged body की एक electric potential energy होती है. So when protons are moving from this point to this point, when this movement takes place, हमारे पास एक electric potential energy create हो रही होगी. That electric potential energy is used to combine ADP and inorganic phosphate to ATP. Is this now clear, Rashad? And to the rest of you? Perfect. And when the protons are moving down the electrochemical gradient, उस process को हम लोग chemiosmosis बुला रहे होते हैं. And chemiosmosis always results in the ATP synthase to synthesize ATP. Now the question here is, हमारे पास ATP बन रहा है, chemiosmosis हो रहा है, हमारे पास जो इलेक्ट्रॉन्स हैं वो भी ट्रांसफर हो रहे हैं. How is this process, or why is this process different from respiration? Why is this ATP production not under the umbrella of respiration? I would like you guys to think what is the definition of respiration. Think what respiration means and then answer. Okay, I'll repeat the question. हमारे पास जो phosphorylation अभी हुई है, photophosphorylation हुई है, that is producing ATP, and that is producing ATP through stock particles through the mechanism of chemiosmosis. Then why is this chemiosmosis or this ATP synthesis different from the respiration process? Very good. So most of you have answered this correctly. हमारे पास respiration की क्या definition होती है? Respiration is defined as the oxidation or breakdown of organic molecules to produce ATP. Over here, the entire process of photosynthesis is being conducted to make an organic molecule. We do not have an organic molecule in the first place. We are making an organic molecule. And because we do not have an organic molecule, and that organic molecule is not being oxidized, is not being broken down to make ATP, that is why this ATP synthesis is not same as respiration. Is this clear? Most of you guys answered it correctly. Very good. अच्छा. Now, हमारे पास there is a question. हमारे पास electrons जो हैं, we understand that the electrons are coming from the chlorophyll pigments, which are getting photoactivated, which are getting excited. But we need this continuous flow of electrons for the continuous photophosphorylation to take place. Now, there has to be constant supply of electrons. हमारे पास electrons यहाँ पर भी तो आ रहे हैं, वो replace कर रहे हैं. We need electrons to fill in the space, to fill in the empty space that is created once a chlorophyll pigment gets excited. So how and why these electrons are coming from? Now, हमारे पास ये ऑक्सीजन इवॉल्विंग कॉम्प्लेक्स है. हमारे पास लाइट के दो रोल्स होते हैं इन रेस्पिरेशन. एक रोल हमारे पास होता है द फोटो एक्टिवेशन ऑफ पिगमेंट्स. बिकॉज फोटो एक्टिवेशन होगी तभी हमारे पास इलेक्ट्रॉन लूज करेगा क्लोरोफिल पिगमेंट और वो इलेक्ट्रॉन ईटीसी पिक करेगा. सो वन रोल ऑफ लाइट इज फोटो एक्टिवेशन. The other role of light is to split the water molecule. And the water molecule would not randomly split. For example, if you have a glass of water and you put it under light, हमारे पास water split नहीं करेगा या वो water oxygen produce करने नहीं लग जाएगा. There has to be a certain condition or there has to be an enzyme to trigger that photolysis to take place. So just under the light, if you place water, हमारे पास oxygen evolve नहीं होगी. There has to be a water splitting complex. There has to be an oxygen evolving complex with which the water molecule would bind. So let's say this is the water molecule. This is water. It attaches or it binds with the oxygen evolving complex or the water splitting complex. Sorry, water splitting center. And once it binds with the oxygen evolving complex and the water splitting center, what happens is the water undergoes photolysis. And as a result of photolysis, we have protons and electrons and half a mole of oxygen. Now, These electrons that the water molecule releases after photolysis, these electrons are replaced in the reaction center, or they're given back to the chlorophyll pigment, and only then this electron transport system is continued. Oxygen is evolved as a byproduct or a waste product, and it can either be used for respiration or it can lose via the stomata. Now, what about this hydrogen? हमारे पास जो ये हाइड्रोजन बनी है, okay, I'll repeat this part. हमारे पास यहाँ तक हमने अंडरस्टैंड कर लिया था that the chlorophyll pigments are getting photoactivated, and because of that photoactivation they are losing electrons, and when they're losing their electrons they are picked up by the ETC, and हमारे पास ETC पे movement होती है, active transport होती है, active transport के बाद chemiosmosis होता है, which results in the photophosphorylation. However, the question is, हमारे पास the ETC movement or the ETC flow has to be continued, right? हमारे पास there has to be a constant supply of electrons, because एक दफा क्लोरोफिल ए ने अपने इलेक्ट्रॉन्स दे दिए, क्लोरोफिल ए के पास इन्फिनाइट अमाउंट ऑफ इलेक्ट्रॉन्स तो नहीं होंगे, उसको तो इलेक्ट्रॉन्स चाहिए होंगे, because even if for example we say, hypothetically speaking, if a chlorophyll molecule has, let's say, 100 electrons, एक पॉइंट आएगा जब उसके सारे 100 electrons खत्म हो जाएंगे, it would donate them, or it would lose them under the process of photoactivation. Then where would more electrons come from? We need a constant electron supply for this ETC movement, for this ETC flow to be continued. So these electrons are coming from the photolysis of water. For the photolysis of water, there has to be an oxygen evolving complex or a water splitting center to be present. The water molecule would attach itself, would bind itself to this oxygen evolving complex. And when it undergoes photolysis at the oxygen evolving complex, हमारे पास it would form, or it would break down into hydrogen ions, into electrons, and into oxygen. Is this now clear? Perfect. Now, हमारे पास ये जो electrons हैं, जो water से आ रहे हैं, ये electrons कहाँ चले गए? They enter into the reaction center, और reaction center से हमारे पास जो ETC का pathway था, that is continued. Now the question is, where and how does these protons come into action? Now, we have an enzyme over here. So here is an enzyme. Now, this enzyme is known as NADP reductase. And from the name of this enzyme, we can figure out that this enzyme would cause the reduction of NADP. So हमारे पास यहाँ NADP present होता है, already present होता है यहाँ पर NADP. The hydrogen ions, or the protons that were produced as a result of photolysis, these protons are accepted by NADP. And you would also notice कि हमारे पास जो ये enzyme है, जो ये NADP reductase है, it is present with an ETC protein complex. So not only does the NADP reductase combines NADP with protons, but it also acts as the final electron acceptor. So जो NADP reductase है, what it does is, it takes the protons which came from the photolysis of water, and it takes the electrons from the ETC to form NADPH. Is this clear? That the final electron acceptor is the NADP reductase. It combines NADP with electrons from the ETC, and it combines NADP with the protons from the photolysis of water. Okay? So the NADP reductase acts as a final electron acceptor. Is this clear? Do you guys want me to repeat this again quickly? Yes, Mutahar? ओके, आई क्विकली रिपीट दिस. हम इसको एक एक करके ड्रॉ करके जल्दी से रिपीट करते हैं. लेट्स से दिस इज व्हाट this is photosystem 2. This photosystem 2 has what? It has accessory pigments भी होंगी और हमारे पास primary pigments भी होंगी. Light is falling upon this photosystem. Now this light causes photoactivation, or you can say it gets the chlorophyll excited. And in this stage of excitation, the chlorophyll lose electrons. These electrons are picked up by the ETC. So there are a lot of ETC complexes. Okay. Now, as the electrons are moving across the ETC, they lose energy. That energy, let's say हमारे पास thylakoid membrane है, the energy that is lost is utilized to actively pump hydrogen ions inside the thylakoid space. Active transport of protons. Because of this active transport, there is a proton gradient established in the thylakoid space. We have stocked particles. Now, we have higher protons over here, higher concentration of protons in the thylakoid space. In the stroma, we have lower hydrogen ion concentration. These hydrogen ions would move through the stocked particle by the process of chemiosmosis. And as a result of chemiosmosis, हमारे पास photophosphorylation होगी. The thing is that once this photosystem has lost its electrons, इसको electrons की replacement भी चाहिए होगी. Now, how are those electrons replaced? हमारे पास यहाँ पर एक water splitting center या oxygen evolving complex होगा. This oxygen evolving complex, यहाँ पर हमारे पास photolysis of water होगा. Water would split into protons, into electrons, and into oxygen. Now, these electrons enter the reaction center and they continue the ETC wali movement. Now, हमारे पास there is an enzyme called NADP reductase. Now, NADP reductase acts as the final electron acceptor and it combines NADP and the water and the protons that came from water together with NADP to form reduced NADP. Is this now clear? Any questions? Yes, exactly, Ishad. So NADP reductase combines NADP with the electrons from ETC and the protons that came from the photolysis of water. Amar, आपको clear है या कोई question है? It's on the thylakoid membrane. All of these things, the ETC, the photosystem 2, the photosystem 1. अच्छा, मैं इस वाले में photosystem 1 draw करना भूल गई. There is a photosystem 1 also present over here. Um, it's near the photosystem 1. It's near the photosystem 1. ठीक है? Is this now clear? Now, अगर हम electrons की movement देखें, we can say that the water gave electrons via photolysis. ठीक है? हाँ, PS1 के पास ऑक्सीजन कॉम्प्लेक्स मॉलिक्यूल नहीं है. बिल्कुल भी नहीं है, टोटली. बट मैंने कहा ना कि हमारे पास हम तो खाली एक सिस्टम देख रहे हैं. ऑन अ थाइलाकॉइड मेम्ब्रेन हमारे पास बहुत सारे फोटोसिस्टम 2s होंगे और बहुत सारे फोटोसिस्टम 1s होंगे, बहुत सारी ये एंजाइम्स होंगी एनएडीपी रिडक्टेस और बहुत सारी एटीपी सिंथेज होंगी. सो वी आर जस्ट सीइंग वन स्पेसिफिक पार्ट ऑफ इट. सो हमारे पास these electrons are passed through ATC as Ahmed mentioned, yes. So NADP reductase can be present anywhere in the thylakoid membrane. ठीक है? Now, if we look at the flow of electrons in this entire story, water split हुआ, उसने अपने electrons दिए. Photo system 2 accepted these electrons, and from photo system 2, these electrons went to the ETC, and from ETC, these electrons were accepted by NADP, or you can say yeah, just NADP भी सही है. So now, if you notice, हमारे पास the starting point and the ending point of this photophosphorylation is not the same. We have a different starting point and we have a different ending point. So this photophosphorylation or this electron transfer is linear in nature. It's not cyclic. Because हमारे पास for anything to be cyclic, हमारे पास starting point और ending point same होना चाहिए. Over here, we started off with the water molecule, and the final electron acceptor was an NADP. So this entire exchange of electrons is non-cyclic or linear in nature, and hence the name non-cyclic photophosphorylation. Now, what happens in cyclic photophosphorylation? Now, in cyclic photophosphorylation, we do not involve the photosystem 2. It is only concerned with the photosystem 1. Now, what happens is this is your thylakoid membrane. This is the thylakoid membrane. This is your photosystem 1. Okay, photosystem 1 does not have any what do you say oxygen evolving complex. तो हमारे पास यहाँ पे कोई oxygen evolving complex नहीं होगा. Now, we have the chlorophyll pigments, chlorophyll A over here. Light energy would fall upon these pigments, causing them to become excited. When they become excited, they lose their electrons. These electrons are accepted by ETC. They move from one ETC to another. Now, in this process, they are losing their energy. That energy is utilized to actively pump hydrogen ions. So, the hydrogen ions are actively pumped, due to which a proton gradient is established. हमारे पास dot particle present होगा. These hydrogen ions would move down the electrochemical gradient. Chemiosmosis होगा. Chemiosmosis would cause the phosphorylation of ADP to ATP. Now the question is, what happens to this final electron? Where does this go? So this final electron re-enters the photosystem 1 and the cycle continues. Now you can clearly see कि हमारे पास यहाँ पर जो starting point था और जो ending point था, that was same. There was a cyclic flow of electrons. And in the cyclic photophosphorylation, only ATP is made. No oxygen is formed, no reduced NADP is formed. There is only ATP made in a cyclic photophosphorylation process. Is this clear? Any questions about cyclic photophosphorylation? The electron re-enters back into the photosystem 1. See, हमारे पास वो electron कब lose हुआ था? The electron was lost when light fell upon the chlorophyll pigments. The chlorophyll pigments absorbed the light. They underwent photoactivation, excitation, due to which they gave away their electron. This electron was picked up by the ETC and it moved across the ETC, and that electron came back to the photosystem 1. So the final electron acceptor is photosystem 1 again. So the photosystem is giving away the electron and it is also accepting the electron back. Yes, the process is like that the electron re-enters photosystem 1. Yes, and Ishal, also in non-cyclic photophosphorylation, both the photosystems were involved. We had PS2 and we had PS1. In cyclic photophosphorylation, we only have this part. ये सब नहीं हो रहा होता in photophosphorylation, that is non-cyclic. That is cyclic, sorry. So in cyclic photophosphorylation, it's only PS1 that we are concerned with. In non-cyclic, we are concerned with PS1 and PS2. An easy way to remember this could be NCP and CP. ठीक है? Over here, NC से आप याद रख लो, two photosystems are involved. And over here, just remember, one photosystem is involved. So NC and C. ठीक है? So in non-cyclic, we have both the photosystems involved, and in cyclic, we only have one photosystem involved. और हमारे पास दोनों cyclic photophosphorilation और non-cyclic, both of them are taking place on the thylakoid membrane. Is this clear? The light dependent reaction. तो हमारे पास अगर हम yields देखें, in a non-cyclic photophosphorylation, हमारे पास oxygen बन रही थी. We had oxygen over here as a result of photolysis of water at PS2, and we were forming reduced NADP, and because of chemiosmosis, we were forming ATP. So there are three things that we are forming in non-cyclic photophosphorylation. We are forming ATP, we are forming water, and we are forming NADPH or reduced NADP. On the contrary, when we talk about cyclic photophosphorylation, we are only making ATP. There is only one product in cyclic photophosphorylation. The purpose of cyclic photophosphorylation is simply to produce more and more ATP. क्योंकि ATP हमें क्यों चाहिए? ATP is required for the light dependent stage. तो जितनी ज्यादा ATP बनेगी उतना ज्यादा हमारे पास ATP होगा for the next stage of photosynthesis. So the sole purpose of cyclic photophosphorylation is to produce more ATP. ठीक है?